In this video we're going to take a look at the neurological control of micturition. Now this is a fairly complex area, however what I'm going to try to do is break it down into a series of stages, and hopefully each individual stage will make sense and we'll be able to put it all together to see how the system works as a whole. So if we're talking about micturition, we really should start off by talking about the bladder. And let's just draw a very simple depiction of the bladder. So here is our bladder, there's the urethra, here's the bladder with its detrusor muscle, and there's the urethra again. The bladder, as we said, has a detrusor muscle with lots and lots of smooth muscle fibres running in all different directions to enable it to empty effectively. It has a bladder neck here, the proximal urethra, and then it has a urethra that passes through the pelvic floor and the prostate if you're a man. The urethra has two sphincters. In both men and women we have an external urethral sphincter which is composed of skeletal muscle and is found within the pelvic diaphragm. And we have an internal urethral sphincter which is only present in men, and the internal urethral sphincter is composed of smooth muscle, and in fact in men its major function is the prevention of retrograde ejaculation, but it does also have a role in continence as well. Now we're going to do an experiment here, a thought experiment at least, and I want you to imagine that we have inserted a catheter into the bladder, and at the end of that catheter we also have a little manometer in order to measure the pressure of the fluid within the bladder. And what we're going to do is infuse fluid into the bladder, measure the volume that we've infused, and look at the changes in pressure that we can measure. And if we do that experiment, which is actually urodynamics type study, we get a plot that looks a little bit like this. So here is a graph with pressure on the y-axis and volume infused into the bladder onto the x-axis. And what we can do is we can infuse increasing volumes of fluid into the bladder and see what happens to the pressure. And we observe something quite interesting. We can actually infuse quite a large volume of fluid into the bladder and actually not get an appreciable increase in the bladder pressure. All right? So we can infuse something like about 150 or 200 millilitres of fluid into the bladder, and the pressure will remain relatively low. However, as we infuse more fluid, we do start to see an increase in pressure. All right, up to about a volume of something like 500 millilitres, where the urge to micturate becomes almost irresistible, and then the patient has to pass urine and micturate. This phase whereby the bladder is increasing its volume without increasing the pressure appreciably is known as the storage phase. Okay, and the storage phase we're going to study on the left hand side of this diagram. So the storage phase is going to be on the left hand side of this diagram, and it corresponds to this graph here. Now when we void, we contract the detrusor muscle, and that expels the urine from the bladder. So the storage phase is the graph going in this direction from left to right, whereas the voiding phase is the graph going in the opposite direction from right to left. And when we measure the pressures within the bladder, we find that we have these spikes of bladder pressure occurring, which correspond to waves of contraction of the detrusor muscle. Okay, so we have these spikes in pressure which correspond to the detrusor muscle squeezing the urine inside the bladder to push it out. And this is the voiding phase, which corresponds to contraction of the detrusor muscle and voiding of urine out of the bladder. Okay, and we're going to depict the voiding phase on the right hand side of our page. So that's the basic events. The storage phase, where the volume increases with minimal increases in pressure until the volumes get very high, at which point we have voiding, where we have contraction of the detrusor muscle, which expels urine out of the bladder. What we're now going to do is look at the neural circuitry which is necessary in order to orchestrate this process. And in order to do that, we need to draw a plan of the entire central nervous system. Okay? So let's just take our time, and we're going to draw a cartoon depiction of the brain, brainstem and spinal cord. So here is the brain, the cerebrum, with its folded surface. Okay. Here is the brainstem. I'm not going to draw the different divisions. I'm just going to draw it as one region. And then here is the spinal cord. And when you draw the spinal cord, make sure you give yourself plenty of space, because we need to put a number of wires inside of it, a number of axons, which are going to be important for regulating this storage phase. So the storage phase is mediated by a relatively simple reflex. And like all reflexes, this storage phase needs to start off with a sensory neuron, the afferent arm of the reflex arc. So I'm going to use the colour red to depict sensory neurons. Okay, so red is going to depict our sensory neurons. And what the sensation is that we detect in the bladder is bladder wall stretch. So within the bladder wall, I'm just going to draw this wiggly line to depict a stretch receptor within the bladder wall. So this is detecting increases in volume in the bladder which lead to stretch in the bladder wall. These bladder stretch receptors are innervated by sensory neurons, which I'm drawing on here. These sensory neurons, like all sensory neurons, have their cell bodies within the dorsal root ganglion, and they're pseudo-unipolar neurons, so I'll depict that like that. These sensory neurons enter the spinal cord at a very particular level, and this is a very important level for all urinary, defecation and also sexual function, because the cord level at which these sensory neurons enter is the sacral cord, specifically the levels between S2 and S4. Okay, and a little way to remember this is that S2, 3, 4 keep the pee and poo off the floor. Okay, so S2, 3, 4 are very important for the regulation of micturition, defecation and sexual function as well. Now these sensory neurons, yes, they enter the spinal cord at the levels of S2, 3 and 4, but in fact they don't synapse at these levels. They ascend to higher levels of the spinal cord. And the levels of the spinal cord that they ascend to are approximately found between the levels of T10 and L2. Now, T10 and L2 is a small part of the cell column within the cord that contains the sympathetic system. Remember that the sympathetics run from something like T1 to L2, with the preganglionics in the lateral horn of the spinal cord, but the sympathetics that have an important role for micturition are only found in the lower half of this region. So these red sensory neurons detecting stretch, which are important for the storage phase of micturition, project up to cord levels between T10 and L2. having projected up to these levels, they synapse upon sympathetic neurons. And this is where we need to introduce a new colour. Sympathetics are blue. All right, so I'm using blue to depict sympathetics. So the sensory neurons synapse upon sympathetic preganglionic neurons within the cord. These are excitatory and they stimulate the sympathetic preganglionics. Sympathetic preganglionics, like all sympathetics, have relatively short preganglionic axons but relatively long postganglionic axons. So here is a postganglionic neuron cell body I've just drawn, and it projects to the detrusor muscle, where it has an inhibitory action on the detrusor muscle. And furthermore, these neurons also project to the internal sphincter, where they have an excitatory action. So what we have drawn here is a simple reflex arc, which has an afferent component, a synapse, and an efferent component. And this is the storage phase which is primarily mediated by sympathetics. To recap, low levels of bladder stretch are detected by stretch receptors, and that information is sent via sensory neurons through nerves into the sacral cord, but these then ascend up to lower thoracic levels. They synapse on sympathetic preganglionic neurons, and these are excitatory synapses which stimulate the sympathetic innervation to the bladder and the internal sphincter. Sympathetics are inhibitory to the detrusor muscle and excitatory to the internal sphincter. And the consequence of this storage reflex is that as the volume increases, the bladder actively relaxes, meaning that the pressure does not increase appreciably. So this is the storage reflex. Now, we've got to add on a few additional components to this system. The first component I want to add on is that the stretch receptors don't just report to the sympathetics. Actually, they also report to the cerebral cortex. And this is how we get a conscious appreciation of how full our bladder is. So we can all at any given time make a fairly accurate appraisal of how full our bladders are, and this comes from information received from the bladder stretch receptors through those sensory neurons. Now, there's another component to this story, and that revolves around the internal sphincter. Now, I said at the beginning that the internal urethral sphincter is actually not a particularly prominent structure in females. So, really, the female urinary system needs to be using a slightly different mechanism, but in fact it's the same mechanism in males as well. And that is that the external sphincter needs to assume more importance here. So there needs to be a mechanism that keeps the external sphincter closed during the storage phase. Now, the external sphincter is made from skeletal muscle, and it is therefore controlled by somatic motor neurons. So in green, we're going to use that colour for somatic motor neurons. All right. And the somatic motor neurons that supply the external sphincter are once again found in the spinal cord levels between S2, 3 and 4. And they project to the external sphincter where they are excitatory on that sphincter. Now, I've not been particularly interested in the particular names of the various nerves that we've drawn here. However, this green nerve that I've just drawn on, supplying the external sphincter, is very important for you to know, because this is running within the pudendal nerve. All right? So the motor innervation to the external sphincter runs through the pudendal nerve, from the sacral cord segments to the external sphincter. Now, what is it then that's controlling the pudendal nerve and the external sphincter? Well, within the brainstem, there is a region which sends axons down to these green somatic motor neurons to keep them active and to keep the external sphincter closed. And these are neurons which are found in a region of the brainstem called the L center. Okay? Now, the L in L center actually stands for lateral, but I think of it more as the leave it a bit center, the center that's important for deferring micturition. The neurons of the L center found within the brainstem send axons down through the cord which serve to stimulate, to excite these pudendal motor neurons. So the L center is important for keeping the external sphincter closed and therefore maintaining continence. So there we have it. That is the storage phase of micturition. And this is very important. All of us at this moment are using the storage phase. All right? However, as we said, as the bladder volume increases, the bladder pressures do start to increase as well. And this will eventually need to result in the activation of a different set of reflexes, those being the voiding reflexes. So now let's move on to look at the voiding reflexes, and then finally we'll look at how these two systems communicate with one another. And what we're going to do is we're actually going to do the same thing on the right hand side of the page. So we need to draw an outline of the central nervous system, and then we're going to add some elements of the peripheral nervous system showing the innervation of the bladder, but this time mediating the voiding response. So once again, draw on your cerebral hemispheres. Draw on your brainstem. And draw on your spinal cord, giving yourself a good amount of space so that you can depict those axons going up and down through the cord. Once again, because voiding is mediated by a reflex, we need to start off with a sensory receptor. And like with the storage response, our sensory receptor is a stretch receptor. It's just the same type of stretch receptor as what we dealt with for the storage reflex. These stretch receptors are innervated by sensory neurons which enter the sacral levels of the spinal cord. So if I put on the cell body of my sensory neuron, I can see it entering the sacral levels of the spinal cord, once again between S2 and S4. These sensory neurons, when we're thinking about the voiding reflex, these synapses upon parasympathetic neurons. Okay, so thinking about the voiding reflex, we need to think about sensory neurons detecting bladder stretch, synapsing upon parasympathetic neurons, which I'm going to depict in yellow. All right, well, orangey yellow, I suppose this is. So parasympathetic neurons I'm depicting in yellow. So here is a parasympathetic neuron, its cell body within the sacral spinal cord. Parasympathetic neurons, as you recall, have long preganglionic axons and relatively short postganglionic axons. So here is the parasympathetic preganglionic. Here is the parasympathetic postganglionic. And what the parasympathetics do is they are excitatory on the detrusor muscle. They cause excitation of the bladder, contraction of the bladder, okay? And the sensory neurons act to stimulate those parasympathetics. So what we've drawn on now is a second reflex arc. Bladder stretch leads to reflex activation of parasympathetics, which leads to contraction of the detrusor. This is the basic fundamental reflex of micturition. This is the voiding reflex. And in fact, this reflex is particularly prominent in babies. If you have had the experience of changing a baby's nappy, you know that babies do not have voluntary control of their micturition, and that they can pee on you with no warning at all. And that's just down to the fact that in a baby, when it gets to a certain level of bladder stretch, it reflexly activates the voiding reflex and causes detrusor contraction and emptying of the bladder. But as our pathways mature and as we start to develop a storage reflex, then we lose this automatic response. Now, let's think a little about this in a little bit more detail, because we've still got to pad out this story a bit in order to explain how the balance can change when we want to void. Now, going back to this sensory neuron here detecting bladder stretch. Actually, these neurons detecting bladder stretch have two modes of firing, if you like. At high levels of bladder stretch, i.e. when we're on this part of the curve, they activate a different part of the brain. So at high levels of bladder stretch, these sensory neurons activate a different region of the brain. So let me just indicate that this is the portion which is activated under high levels of bladder stretch. And when these are activated, they stimulate a region of the brain known as the M center. This sits medial to the L center, and this is the micturition center. Okay, so here is the M center here. All right. And what the M center does is, well, one of its roles is to send excitatory inputs down onto the parasympathetics. So, high levels of bladder stretch not only directly stimulate the parasympathetics at the level of the cord, but they provide a further element of stimulation via the M center in the brainstem, which further excites the parasympathetics. So this is a positive feedback loop, all right? This is a positive feedback loop. And whenever you see a positive feedback loop, you're usually thinking about a binary decision that the body has to make. You've seen positive feedback loops, for example, during parturition with oxytocin, where you're either giving birth or you're not giving birth. In this case, we've got a positive feedback loop within the brainstem and spinal cord causing micturition. Either you're micturating or you're not. So positive feedback loops, we tend to see them when there is a binary decision to be made. However, this M center is not autonomous. The M center is not autonomous. We do have conscious control of it. And this conscious control descends down from the cerebral cortex, and it descends down from a medial portion of the cerebral cortex here called the paracentral lobules. We'll talk about those a little bit more in nervous system, but the paracentral lobules are sitting quite close to the central sulcus in the medial aspect of the cerebral hemisphere. Now the paracentral lobules send both excitatory and inhibitory influences down onto the M center. So you can imagine, can't you, a situation whereby you can feel that your bladder is kind of full, So this pathway is reporting that the bladder is fairly full. You see a toilet, you go to the toilet, and then you're able to strongly stimulate the M centre, causing emptying via the voiding response. Alternatively, you can feel that your bladder is full, however there is no toilet in sight, so you can send strong inhibitory signals down onto the M centre, which hopefully will defer voiding until a convenient time. So this pathway here, going from the cortex down to the M centre, is where our voluntary decision is made about whether to void or not. However, some of you, the more perceptive amongst you, might have noticed a bit of an issue with this system. Because when we get to a point where we're starting to activate the voiding reflex, is it not the case that still the storage reflex is active as well? Yes, this is absolutely true. So these two systems are effectively fighting against each other. How can we resolve this? Well, we can resolve this by adding a further set of connections which allow the voiding and storage responses to communicate. The M centre sends projections to the L centre, but these are inhibitory. So the M centre is able to inhibit the L centre. What does this mean? It means that when you decide to void, when you're at a toilet, you strongly stimulate the M centre. This activates the parasympathetics, causing detrusor contraction, but furthermore inhibits the L centre, which results in relaxation of the external sphincter by switching off the pudendal nerve. So you don't have a situation where the voiding reflex is fighting against the storage reflex. So that's all I want to say about the basic physiology of micturition from the neurological point of view. Thank you for listening.